Now to the last part of the third lecture series on the discovery of the inheritance mechanism and understanding historical thinking as well as different types. So Mendel discovered a type of inheritance, and it's known as Mendelian inheritance or monogenic inheritance. Later, we discover a number of different types of inheritance, ones that are polygenic, meaning many genes for one physical trait. We know ones that are linked to sex chromosomes. We have ones that are codominant, meaning you can have two dominant alleles in an allele model. So it's a 3 allele model as opposed to a 2 allele model, and we'll work on it in class. It's a great classic example using the ABO blood group, different blood types. And then there are other types of phenomena like pleiotropy, which we'll cover, which you'll notice is when you have one gene that's responsible for multiple traits. Let's talk about and refer to the human genome. When you hear the term the human genome, this is it, right? Your human genome contains material, right, or DNA, genetic code, nuclear DNA, right, and you also have helper molecules that are in your mitochondria, and those are part of your mitochondrial DNA. If you were to parse up the nuclear DNA, your nuclear DNA is made up of 46 chromosomes in 23 pairs. You have 22 pairs of autosomes. These are the chromosomes, all of them except the sex chromosomes. The sex chromosomes are different, and they determine the sexual characteristics, the secondary ones that we can oftentimes observe in the skeleton of an organism. The binary biological sex designation, let's be clear about that. Mitochondrial DNA typically is very uniform and similar. There's only one very small hypervariable region that's segmented here, that's referred to as HVS. There's more variability in nuclear DNA because it's larger. You'll notice mitochondrial DNA is only 16,569 base pairs. Nuclear DNA is, billions of base pairs. We move on to describing Mendelian traits in humans. Mendelian traits or monogenic inheritance means that you have one locus that is responsible for one physical trait. The traits that Mendel was using with his P's were all Mendelian or monogenetic, meaning one gene is responsible for one physical trait. Mendel wouldn't have been able to make his conclusions if he was using a model organism and traits that were polygenic, meaning many genes or loci, plural of locus, responsible for one physical trait. We'll talk about other polygenic traits. But Mendelian or monogenic traits in humans are very few by comparison. There are only probably about 5,000 Mendelian traits in humans. Most of our traits are actually polygenic. But I can give you a few examples. So Mendelian meaning that one gene is responsible for a physical trait, or one locus on a chromosome responsible for a physical trait. Here are some Mendelian traits in humans with some classic Hollywood stars. A cleft chin is a Mendelian trait that we see follow a Mendelian pathway. It's dominant, so the expression of it is the dominant state. That means that we can look at this individual with a cleft chin and know that they have at least one dominant allele. We don't know if they are homozygous dominant or heterozygous. We do know that they're expressing the dominant trait. You can look at other individuals that have a widow's peak. That's dominant, right? Heterozygous or homozygous dominant. You notice even the variation in the earlobes. Some individuals have earlobes that are integrated to the side of their face or the side of their head. And that's recessive, meaning you have to have two copies of the recessive allele. If you look at Fred Astaire here, he has earlobes unattached that are kind of flapping in the wind. Those are dominant humans, meaning you have to have only one copy to express that trait. Mendelian traits make it easier to kind of look at lineages. It makes it harder with polygenic traits, and I'll explain that to you and why. And even oftentimes we look at the complex polygenic traits in humans as Mendelian, but they are not. Here are some examples of being able to predict the offspring of maybe children. So I have a number of famous people's, famous persons' genotypes, famous couples like Tom Brady and Giselle here. Tom Brady has a cleft chin, so he has to be either homozygous dominant or heterozygous. Giselle doesn't have a cleft chin, so she has to be heterozygous. And that means that if they have children, their biological offspring are either going to be a mix of children with cleft chin that are certainly heterozygous, because they could only get the recessive allele from Giselle, or they won't have cleft chins at all, and then we would also be able to reveal that Tom Brady is in fact heterozygous, because that's the only way their offspring can get 2 copies of the recessive allele and not express the cleft chin. Let's look at David Beckham and Posh Spice, Victoria Beckham. The earlobe unattached trait. David Beckham has earlobes that are attached. And because we know this follows a Mendelian pathway, he has to be homozygous recessive, because you have to have 2 copies to express this. Here you have his wife, Victoria Beckham. She has earlobes unattached. That means that because it's a dominant trait, that she either has to be homozygous dominant or heterozygous. Their offspring will give us an indication maybe of their genotypes. But those are the potential offspring and predictions. The last couple here, Ryan Reynolds and Blake Lively. A widow's peak. A widow's peak is dominant in humans. And so by observing the widow's peak on both of them, they have to either be homozygous dominant or heterozygous. That means that all of their children are going to have widow's peaks, because they are only contributing, right, the dominant allele if they're both heterozygous dominant. However, think about this. We can observe Ryan Reynolds and Blake Lively's children. If they are actually homozygous recessive, meaning they aren't expressing the widow's peak, then we be able to tell that Ryan Reynolds and Blake Lively are both heterozygous for the widow's peak allele. There are a number of other Mendelian traits in humans that are detrimental to the organism. However, at the same time, they may provide benefit in some environments. And because we have culture and compassion, right, some of these traits persist. Some of them are not a detriment, and some individuals can live completely normal lives. Here, now that you have some background in this course, you can see new discoveries made and understand things like autosomal dominant and autosomal recessive in a Mendelian sense. Autosomal meaning it's not in the sex chromosomes. It's got to be chromosome 1 through 22, at least in humans. Dominant meaning you only need 1 to express that trait. Here's an example of a trait, brachydactyly, short fingers. That is autosomal dominant. You only need one allele to express that trait. If something is autosomal recessive, it means you have to have 2 copies. Albinism is an example, or lack of pigmentation. In the autosomes 1 through 22, and you have to have 2 copies for it to be expressed. When we see genetic disorders or other diseases develop, ones that are very virulent and deadly are ones that are autosomal dominant, meaning you only need 1 copy to express it. Sometimes autosomal recessives you'll have carriers, but then you won't run into the disease as frequently because you need 2 copies to express it. Other traits follow a pathway that doesn't include the autosomes, and they're actually sex-linked. And so we have sex-linked traits, ones that are Y-linked. Y-linked traits are ones that females don't get. So if it's Y-linked, it's a male trait by default. There are some traits that are X dominant, X recessive. When you look at females, right, female pathway. However, males still have an X chromosome, if you can recall. So something that's X recessive in females means they actually have to have 2 copies to express it. X dominant in females means that they have to have only 1 copy to express it. In males, if it's X dominant or X recessive, if you get one that has that expression, you either get a gradient of it. One that's not as one that's mild if you have the recessive allele, and one that's a little more intensely expressed if you have the X dominant allele. In males, when you look at the expression of X dominant, X recessive, it's known as hemizygous, meaning that it's expressed in them even though 1 copy is present. So you can see sex-linked traits, males are highly biased towards them, because if it's Y-linked, they're the only ones that can get that trait. If it's X-linked, they have the dominant or recessive trait, it's going to be expressed. Here's an example of a sex-linked trait within the royal family, hemophilia. You can see that hemophilia is an X-linked trait, and you'll notice that there are a number of females here that were carriers for hemophilia. So you'll notice that the boxes represent, or squares, males, and the circles represent females. For a female to be a hemophiliac, she needs to have both copies to be present, right? So it's X-linked recessive. You'll notice that none of the females in this lineage are actually had hemophilia, right? The females, like Queen Victoria, were the only ones that were carriers, meaning they only had 1 copy, but the trait was not expressed. That's why you notice that everyone that actually got hemophilia, which is a condition that doesn't allow your body's blood to clot properly, and you bleed a lot, almost to death. 1 cut could actually kill you. And you'll notice that it's only the males that actually expressed it, right? And because no female acquired 2 copies of it because it's recessive. And you'll also notice, look at the longevity, the life expectancy of males that were hemophiliacs was still very low. Another phenomenon that we notice is codominance. Codominance is where you have 1 or more alleles that are dominant. This is how you can have AB blood, because A is the dominant allele and so is B. So I'll introduce you to the AB blood group here. You might be familiar with this. And the O is recessive. The A and B, in terms of codominance and actually what A and B is, A and B, they are antigens, and they are in the surface of red blood cells. This is a protein that is on your red blood cell. If you have the O allele, you don't have the antigens. You don't have the proteins. They're absent. And so we can look at the different genotypes and phenotypes based on the ABL blood group. So if you have A or B blood, you are either homozygous dominant, meaning you have both A alleles or both B alleles, or you could have 1 A allele and 1 O allele, and it's because O is recessive and A is dominant. You will have A or B blood, and of course there are 2 dominant alleles, A or B. This is also why you can have AB blood, meaning you have 2 copies of the 2 different dominant alleles. If you have type O blood, you are homozygous recessive. Polygenic inheritance is quite fascinating, but it's also really complex. It is non-Mendelian in nature, it is not discrete, and it forms a continuous gradient of expression. It's non-Mendelian, meaning that it takes more than 1 gene that's responsible for one physical trait. So it's many genes responsible for one physical trait, and usually what you'll see, like this picture, you'll have like a distribution, like a normal distribution or bell curve where most individuals fall into with other extremes. This is a continuous range of expression. There's much more variation. When you look at a graph of Mendelian traits, it's like a bar chart where it's like you either have it or you don't. And so most traits in humans are polygenic, and that makes it a little difficult to study. So here are some examples: height, skin, eye color, hair color. These are all examples. Body mass are examples of polygenic traits in humans. I think the last study investigating height, they suggested that there are about 150 to 180 loci responsible for human height. That's enough. That's quite a few genes. The last concept I want to impart on you is pleiotropy. And this is a phenomenon that we notice when one locus or one point on a gene is responsible for multiple traits or features. The example I usually like to give are, light-skinned or albino cats. The same area that's responsible for the pigmentation of their fur or hair is also responsible for their ear. And so what you'll notice is that cats that lack pigmentation in their eye and their fur, that gene is also associated with the development of their ear. And so if there's very prominent albinism and lack of pigmentation, you also have a premature ear and not quite functioning, and meaning that they are deaf. And so most white cats that have blue eyes are deaf. And if they have a yellow or a brown eye, typically there's more melanin production in there for the iris of their eye, and then that also allows for a greater production of the mechanism to help them hear. So most cats that are albino and white that have blue eyes, they can't hear or are deaf. If they have a brown eye or two of them or a yellow eye, they can usually hear much better. If they have one brown eye or yellow eye and one blue eye like this, usually this cat can only hear in its right ear and is deaf in its left. So you may even have expression on different sides of the body.